All right, so nervous system. Let's talk about this. So this is just a fascinating area, and it is an area that we've only now just begun to develop some sort of understanding with. And I want you guys to make absolutely no mistakes. Our understanding of the nervous system, of the brain, of how things work, particularly of consciousness, is extraordinarily limited. Okay, so don't let anyone fool you into thinking that we have a decent understanding of this, because we don't. This is one of the least understood areas of physiology, and this dysfunction of the nervous system leads to very non-intuitive problems and very pronounced problems when we talk about seizure disorders, for example, or we talk about dementia or delirium, or talk about damage to certain areas of the brain from strokes or injuries or metabolic derangements. We see these really strange and non-intuitive things occurring. So I'm going to start taking an up-down approach. So we're going to start at the big picture, and then we'll narrow down to the small picture, and then we'll start at the small picture and work our way up and try to develop some sort of intuition of what's going on here. So let's look at the big picture. So what is a nervous system? Well, it's more or less the command and control center for all body functions. Okay, and there is a computer-like analogy. Now, this is only an analogy, and it fails in many important ways, but it's a good basis. Perfect. We knew that was going to happen, right? And you're still surprised. Perfect. All right. Well, everybody's awake now. Awesome. If I had my druthers, I wouldn't put the chairs up, but that's not my thing. Take that up with choice. See how far you get. You won't get very far at all. Okay, so you can think of the nervous system as kind of like the central processing unit, the CPU of a computer, right? Or really the CPU and the hard drive, because we know the hard drive is where information is stored in a computer, and then the CPU, right, the central processing unit, is where all of the integration of information happens, right? Input, output, and then storage occurs in the hard disk. And so you can think of this as roughly analogous to the nervous system. So what are the major functions of the nervous system? The major functions include receiving sensory information. So information comes into the nervous system, okay? And then the nervous system processes and integrates that information, makes sense of that information, and then when it does make sense of that information, it sends motor information out. So you have sensory information coming in, getting integrated and processed, and then motor information gets sent out in response to that. So if you cannot get sensory information into the nervous system, will anything happen? No, nothing at all, right? So you could not take the nervous system out of a human being and kind of isolate it like a brain in a jar kind of situation. That really wouldn't work, because you wouldn't be able to get any information into that. Does that make sense? So for things to work, the nervous system has to be wired into your body, so to speak. And it has to receive input, right? It has to get information coming in, so information coming out. And remember, we talked about drugs like induction agents and respiratory, like ketamine, for example. Well, that's the basis for how many of these induction agents work, is they block the ability for information to get into the brain, right? They prevent sensory information from getting into the brain. So once the brain quits getting input, what happens? It doesn't have any information, right? And so there is really nothing going on, so to speak. At least at a conscious level, you're still getting information about, unconscious information about how your heart's beating and your blood pressure, and right? And some of those lower brain centers continue to work. But even then, could you give somebody a high enough dose of an anesthetic that you could prevent information from going into those centers? Yeah, you could. And you can actually kill somebody with anesthesia. You just completely shut down their nervous system with a high enough, you could do that with a high enough dose of morphine or fentanyl, as you guys probably have seen, right? You've run into people who are near death because they've taken enough of something that shuts this whole process down. Okay, so the major components of the brain, of the nervous system, of the central nervous system include the brain and the spinal cord. The brain and the spinal cord make up the central nervous system. And then the part of the nervous system that branches away from that, either directly out of the brain or directly out of the spinal cord, that's all known as the peripheral nervous system. And there are two ways of getting in or out of the central nervous system. Okay, there are two ways. There are what are called spinal nerves, and these are nerves that go in and out of the spinal cord. And then there are also what we call cranial nerves, and these are nerves that go in and out of the brain directly. Okay? So there are nerves that connect directly into and out of the brain. Those are called cranial nerves. And then spinal nerves come in and out of the spinal cord. Does that make sense? Now, both the cranial nerves and spinal nerves are considered the peripheral nervous system. You guys cool with that? All right, cool. So central nervous system, the CNS, the brain and spinal cord. Peripheral nervous system, the PNS, cranial and spinal nerves. Now, there are two major pathways, okay, nervous pathways. They're what we call afferent pathways with an A. Afferent means to ascend. So these are pathways that bring information into the brain. So if I'm bringing information into the brain, that is known as sensory information. Sensory information ascends into the brain. Are you guys okay with that? Okay, these are sensory pathways that bring information into the central nervous system. Efferent pathways are also known as descending or effector pathways. And these are almost always motor information pathways leaving the brain and going out to organs, muscles, tissues, glands, etc. So, afferent to ascend sensory, efferent descend motor. You guys go with that? For example, I have this coffee here, right? This is my old man's coffee. It's decaf with a sugar-free sweetener and stevia in it, right? I'm so metropolitan, I'm losing my edge, right? So sensory information is coming into me. I'm seeing this. Ooh, it's red. Ooh, red, pretty, right? Shiny stuff, right? So that's coming in through an afferent pathway, i.e. my optic nerves, right? My optic nerves are relaying that information. That information is going into the occipital lobe. It's getting integrated and processed, and then it's being shipped out to other areas of my brain, right? And then there are areas that interpret that information as, oh, I'm thirsty and I'd like a taste of that sugar-free coffee there in front of me. And then efferent motor information is sent out to my arm to make me grab this and to drink that. And that's the general mechanism by how this all kind of works. We also have what are called interneurons. Okay? Interneurons are bridging neurons. They bring information from one neuron to another, so they act as a bridging mechanism between the afferent and efferent pathways. Does that kind of make sense? You have afferent pathways, efferent pathways, and you have these interneurons that interconnect that, right? That kind of makes sense, doesn't it? Okay, so somatic, soma means body. Somatic sensory pathways bring sensory information in from the skin. Okay? So somatic pathways begin with receptors in our skin, right? And you guys remember, did you study the different types of pain in the intro class for advanced assessment? Somatic versus visceral pain, right? Somatic pain tends to be sharp, right? If you cut your hand, that's a kind of a sharp pain. And it's localized generally, right? You can point exactly where that pain is. It's sharp and local. Visceral sensory pathways, however, bring sensory information in from organs. And viscera means organ, right? And we know that visceral pain, is it typically very sharp and pinpoint? No, it's very dull, crushing, aching, squeezing, and it's very diffuse, and in some cases may even be referred, or may spread or go to other areas of the body, right? So that's visceral versus somatic. Okay, so let's look at the major divisions of the nervous system graphically. So you've got the CNS, the brain and spinal cord, and you've got your afferent pathways coming in, your efferent pathways going out. So far, so good? Now, the central nervous system gets information in and out through the peripheral nervous system. You guys okay with that? Now, there are two major branches of the peripheral nervous system. There is what we call the somatic. This is also known as the voluntary branch of the peripheral nervous system. Why is it voluntary? Because we have control over it. For the most part, we have conscious control over it. So the somatic nervous system primarily includes skeletal muscle, right? We have primary control over skeletal muscle. And then there is the autonomic branch of the peripheral nervous system. And I think of the autonomic as being somewhat automatic. It's not entirely automatic. We do have some conscious control over parts of it, but by and large, what occurs with the autonomic nervous system is subconscious. We don't need to put a whole lot of thought into it, okay? Right, like digestion, right? Like information going to your heart and your lungs, right? That kind of stuff. Your mucus secretions, gastric secretions, right? By and large, we don't have conscious control over that. There are some techniques and some things that some people can do to exert some degree of control over that. So I'm being very general when I say that, but in general, this is a good foundational way of looking at it. And then we have two branches of the autonomic nervous system. We have the parasympathetic and the sympathetic. All right, are you guys okay with that, sympathetic versus parasympathetic? Sympathetic in general tends to be the quote-unquote fight or flight response. So this is the branch of the autonomic nervous system that is... Am I in the way?
Oh, no.
Oh, okay.
No,
Okay.
I was just trying to get...
Okay, cool. Sorry. This is the branch of the peripheral nervous, autonomic, which is part of the peripheral, that speeds our heart rate up, increases respirations, increases blood pressure, shunts blood away from the periphery into our core, prevents us from digesting, opens our lungs and our airways up, right? This is the part of the nervous system that has evolved for minute-to-minute survival. Does that make sense? This is our immediate survival mechanisms. Now, the parasympathetic nervous system is a little different, right? The parasympathetic nervous system, some people refer to this as the feed and breed, right? So this is responsible for slowing things down in some areas, slowing our heart rate down, right? Decreasing our blood pressure, not shunting blood to our vital organs, but allowing blood to go to non-vital organs, primarily our digestive tract, right? So we can absorb nutrients. Increasing secretions, right? This is the branch of the nervous system that causes increased mucus secretion, increased gastric acid secretion, increased digestion, right? So we see a lowered heart rate, a lowered blood pressure, increase in digestion, and so on and so forth. You guys okay with that? Now, some people say that the sympathetic nervous system is like the gas and the parasympathetic nervous system is like the brake. That is not a good analogy. Please do not look at it that way, okay? These are not necessarily opposing each other. To normally function, we need to have a balance of parasympathetic and sympathetic. So you need to have a certain amount of parasympathetic tone and a certain amount of sympathetic tone. And so what I think of is the sympathetic is like putting your foot on the gas, and the parasympathetic may be like letting your foot off the gas a little bit, but not hitting the brake necessarily. Hitting the brake would be like blocking the sympathetic, and the parasympathetic doesn't really block the sympathetic. That's not how it works. These things work together at the same time, and sometimes you have more sympathetic tone, and other times you have more parasympathetic tone. You guys cool with that? So far, so good? Yeah, sludge would be a over-exaggerated parasympathetic response. Yeah, for sure.
And it hides the effects of the sympathetic.
Yes. Or in some, yeah, it does. Or in some cases, you can actually disconnect the sympathetic nervous system. For example, in spinal shock, right? If I have a spinal injury that cuts the sympathetic nervous system off, because the sympathetic nervous system primarily, and we'll talk about this here in a little bit, it primarily comes out of the central nervous system at the spinal cord, typically thoracic and lumbar spinal roots. That's your sympathetic nervous system. Your parasympathetic nervous system comes directly out of your brain. It's a cranial nerve called the vagus nerve. The vagus nerve is a primary parasympathetic nerve. So if you have a spinal cord injury, you could cut the sympathetic nervous system off, but still have a functioning parasympathetic nervous system. So what happens is all the sympathetic tone goes away, and all you have is parasympathetic tone, and so that's why the blood pressure gets low, the heart rate gets low, they vasodilate out, and they have kind of this pink warm skin, and that's why they look so weird. It's not that the parasympathetic nervous system is inhibiting the sympathetic, it's that something cut the sympathetic off, and all you have is parasympathetic. Does that kind of make sense? So yeah, what's that? What's that? Well, negative feedback would be something inhibiting something. So like the renin-angiotensin-aldosterone system would be a great negative feedback. Here, there are negative feedback systems, but it's not between the parasympathetic and sympathetic per se. There are other mechanisms, other feedback mechanisms that occur. But there's definitely negative feedback that underpins both sympathetic and parasympathetic, yeah, for sure. Okie dokie. Moving right along. All right, so now that we've taken a look at the big picture, what I want to do is I want to take a look at the small picture. All right. So I want to ask you guys, what is the functional unit of the nervous system?
Nucleus.
What's that?
Isn't it the nucleus?
Well, the nucleus is the center of a cell that contains genetic material. Yeah, the neuron. The neuron, okay. The neuron is generally what we consider the functional unit of the nervous system. Yeah, the neuron. Neuron literally means nerve cell, right? Nerve cell. Okay. So, these are the functional units of the nervous system, the neurons. And here I have a Rainier's gold chloride stain microscope slide that I took a picture of. And you can see these neuron cell bodies, and they kind of have this little star shape there. They've got a nucleus. Right? You guys see that there? Okay, that's that classic neuron. We'll talk about the neuron anatomy, physiology here in a little bit. And here's an interesting analogy. We have approximately, give or take, several million, but we have approximately 100 million neurons in our brain. There are approximately 100 million stars in the Milky Way galaxy. So if you think about it, you have a neuron for every star. You have a galaxy of neurons in your brain. That's pretty remarkable. And then what's even more remarkable is, on the average, each neuron has about 10 to 20,000 connections with other neurons. So when you guys took anatomy and physiology, you probably remember the neuron with its axon connecting to another neuron, right? You had that basic picture. Well, that's not exactly the whole picture. It is 20,000. Yeah, each neuron, on average, again, give or take, but, 10, 20,000 connections.
Still only one axon coming out of it, though, right?
What's that?
One axon coming off. each
So yeah, you have an
branches
axon, but
off.
then you have your axon terminals that branch out. Yeah, yeah. So that's incredible. So you have trillions and trillions of individual connections. And so hopefully it's starting to make sense why we don't have a great understanding of this, because this is just so incredibly complicated when you think about this. Okay, so neuron is a functional unit, but the neuron is not the most numerous. That's what's even more incredible, is the neurons aren't the most numerous cell in your nervous system. That belongs to the group of cells known as the glial cells, or the neuroglia. Glia, A is plural, right? Glia, or gliae, right? And the glial cells help the neurons. They are support cells for the neurons, to protect the neurons, to help them in their day-to-day lives, quote unquote. You have two general types of glial cells. You have what are known as your microglia and your macroglia, all right? And the microglia are very small, and these are nervous system macrophages. So these are immune cells. And is there another area of a body that have immune cells embedded in it that we just got done talking about in the last block, perhaps?
Isn't the alveoli, don't they?
Perfect. The alveolar macrophage, right? You have macrophage within the alveoli. You also have macrophage throughout your nervous tissue, and pretty much you have macrophages throughout all of your tissues. All right? And then the macroglia is a large group of glial cells, and I'm going to talk about some of the more important ones. Okay, so we have what are known as astrocytes. Okay? Astrocytes are the most abundant, not neurons, but the most abundant cells in the nervous system, in the central nervous system, right? And the astrocytes kind of look like little stars, star, right? And they have these little projections that link neurons to the blood supply. And there is this concept in neurophysiology known as the blood-brain barrier. You guys familiar with that term from anatomy? And the blood-brain barrier is basically an extra barrier between the blood and the neurons. So in normal tissues, you have your vasculature, right? Your capillary network coming through, and it's basically just one layer of squamous tissue in the capillaries that separate the capillaries from the tissue beds. And so that allows stuff to easily diffuse in and out through the capillaries. But in the brain, you have another barrier, and part of that barrier may be formed by these astrocytes. There is a little bit of discussion about what's really going on there, but the classical way we understand this is that these astrocytes kind of form another barrier. So what does that mean? Well, that is an extra way of regulating what gets in and out of the brain. And so that actually makes it difficult for us when it comes to treating brain problems, because it is more difficult to get drugs into the brain, particularly like antibiotics. So you have an infection or an abscess in the brain. Getting an antibiotic in the brain can be really hard because it has to get past the blood-brain barrier. Treating brain cancers can be very difficult, in part because it is so difficult to get chemotherapeutic agents into the brain to work, right? So this blood-brain barrier is very protective, right? It helps the brain have hyperregulation of what goes in and out, but it is also a hindrance when it comes to treating brain disorders. And we often have to develop special drugs, prodrugs, to get into the brain. For example, a common Parkinson's drug is levodopa and carbidopa. You guys familiar with those? Carbidopa, levodopa, those are classic. And those are dopamine precursors, because part of what happens in Parkinson's is a dopamine deficiency due to loss of cells in certain areas of the brain, dopaminergic cells, neurons, in motor coordination areas of the brain, substantia nigra and other areas. And one way that can help is by increasing dopamine levels. Well, you might think, well, why don't we just put somebody on a dopamine infusion, right? That's easy enough, right? I mean, we all have dopamine. We all know how to give it, right? Why don't we just do an infusion of dopamine? Well, guess what? If I give you dopamine, I infuse dopamine into your blood vessels, that dopamine cannot get into your brain. It cannot cross the blood-brain barrier. So what we have to do is we have to create special dopamine precursor molecules like carbidopa, for example, that can get into the brain, and then it is converted into dopamine once it gets into the brain, right? And so that's just one example of kind of some of the obstacles that we run into getting stuff into the brain. Okay, so these astrocytes protect, regulate chemicals, even regulate vascular tone, right? So they're protective and regulatory, and as they help the neurons. You also have what are known as oligodendrocytes. And oligodendrocytes form what is known as the myelin in the central nervous system. And you guys know what myelin is, right? Myelin is kind of like an insulator, right? That helps neurons transmit signals more efficiently.
Like a bubble around?
Yeah, it forms a sheath, yeah. And maybe some of you thought that the neurons and cells form their own sheaths. That's actually not the case. It's
individual?
Yeah, it's the oligodendrocytes. It's a glial cell. It's not the neuron at all.
Oh.
The glial cells, the oligodendrocytes, form the myelin in the central nervous system. Okay? In the peripheral nervous system, a different kind of cell forms the myelin. And these are what are called Schwann cells. And they're actually tumors that originate from oligodendrocytes and Schwann cells. There's a, I wouldn't say common, but there is a kind of tumor that some people get, and it involves the vestibular cochlear nerve. It's called an acoustic schwannoma. It occurs in the myelin, not the oligodendrocytes, because it's in the peripheral nervous system. And then we have what are called ependymal cells. And ependymal cells produce cerebral spinal fluid, or CSF. And we know what CSF is, hopefully, right? Right? CSF acts as kind of a cushioning, protection. And you have CSF inside of your central nervous system, right? And outside. So it kind of acts as like your brain kind of is floating in it, inside and outside, and it acts as kind of a protective cushioning barrier, if you will. And these ependymal cells actually produce a CSF, and you produce about 500 milliliters of CSF per day. It's kind of a lot if you think about it. So you're constantly producing it and then reabsorbing it. And it's actually produced in a structure known as the choroid plexus. And we'll talk about the ventricles, where you have it inside, and the meninges where you have it outside of the brain here in a little bit. Okay, so you guys good with that? So your neurons are the functional units, and then the glial cells are the support cells. All right, and the glial cells are every bit as important as the neurons. Right, you need both of these for your brain to work. And then finally, you have what are called satellite cells, and these are just involved in quote unquote, involved in regulating the external environment, acid-base balance, chemicals, molecular movement, those kinds of things. Okay, so you've got your neurons, and you've got your glial cells, and then you have all different types of glial cells, and you have different types of neurons as well. So far, so good? All right, move right along. So let's talk about the neuron. Like we said, the neuron is the functional unit of the central nervous system. All right? And we know that neuron activity in specific areas of the brain, the central nervous system, correlates strongly with different types of sensory and motor activity. And the reason, or how we found this out, is there is a relatively new technique known as fMRI. Have you guys ever heard of that term, fMRI? It stands for Functional Magnetic Resonance Imaging. And what we can do is we can take somebody and we can put them into an MRI machine. And then we can have them think about certain things or even do certain motor tasks. And then what will happen is Circulation, that's primarily what fMRI looks at, is circulatory changes. Circulation will increase to areas of the brain when people are doing certain things, playing instruments or thinking about something or having some sort of experience or doing some motor activity, and then those areas of the brain will light up under the fMRI, indicating that there's increased blood perfusion to those areas. And we know that neurons are highly metabolically active, right? They need a constant supply of oxygen, constant supply of glucose to really function. And so when we see that, that correlates with neurons in that area of the brain are generally responsible for whatever sensory or motor activity is going on. Does that kind of make sense?
I'm guessing it follows the iron and the hemoglobin in particular?
No, actual perfusion. Sort of. It's actually looking at actual perfusion, and the way MRI works is very, very, very weird. And you'd have to actually understand a little bit of quantum mechanics. And there's this concept known as spin. Anyone ever heard of that, spin? And you will basically with MRI, you use a strong magnetic field, and you align the spin of certain atoms in a specific way. And then what you do is you send radio pulses in, and you kind of perturb that spin. And then as the spin realigns with the magnetic field, radio waves are released, and those radio waves are detected, and that's what produces the image. So it's really, really complicated kind of technology. And of course, it's the same technology that's used in conventional MRI scanners. We call magnetic resonance imaging. If you get an MRI on somebody, it's the same technology, but fMRI is kind of a real-time thing that we do, whereas when you get just a regular MRI, you sit, it does a scan, and then you have static pictures that you look at, right? Static. This is kind of a real-time that looks at blood flow. Oftentimes we use contrast. We'll inject contrast in, and it's the actual contrast that's getting picked up. And as perfusion increases to certain areas, the contrast goes to certain areas and it lights up. Yeah. So does that kind of make sense a little bit there? Yeah. So this has been one of the most important tools that we've developed in the past few decades to really figure out what's going on with the brain. All right. So, what is the neuron's role in consciousness? Boy, this is a big question, right? Consciousness. This is kind of the quote-unquote lights are on, right? Our subjective internal experience. This is a really, really tricky question, and what's going on there? There's a lot of philosophical discussion around this, and obviously we're not going to dive into that in this class here, but there is a lot of, are we maybe just zombies? Is consciousness just not even a real thing? Are we just kind of running a program, and it looks like consciousness, and right, and you talk to somebody and they say, oh, but I have this experience and I feel this and that, and it's all just kind of an illusion, maybe there's something to it. Who knows? I don't know. It's a real hard question, but certainly there's something weird going on, and there's this concept known as qualia. You guys ever heard of that term, qualia? And qualia is kind of this just weird, subjective, like, what is this here, this color? Okay, so those of you who aren't colorblind, this is red, right? Everybody identifies this as red, but this is weird. It looks red, right? But does this red look the same to you as me? Are you experiencing this the same? And how would we know? Like, how could you tell me, well, it's red, everybody knows. But maybe yellow is red and green is, right? And maybe if I were to somehow look through your eyes, it'd look different, the color. We don't know, right? And that's just one example of this weird rabbit hole we can go down when we talk about the qualia, right? It's a weird thing to think about, right?
It's a conversation that requires copious amounts of marijuana.
Well,
He cannot say.
yeah, well,
Oh, that's awful.
okay, yeah,
Okay.
yeah, that's maybe a conversation we have outside of class, but there's certainly, yeah.
5 o'clock, my
Yeah. There's certainly, yeah, turn off the recorder, right? No, but as you imagine, this is a really weird area, and I'm certainly not well prepared to talk about it, and people much more intelligent than I are grappling and struggling with this. But here's a general picture. We can't explain how we go from a neuron to consciousness, but here's what's going on with neurons. Okay, so you guys know the general structure of a neuron, right? You've got the soma, which is the cell body. It contains the nucleus. And you have these projections that bring information into the soma, and these are known as the dendrites, right? The dendrites bring information into the neuron, and then information gets processed in the neuron, okay? And you have a very important part of that's known as the axon hillock there, right? And you actually have these little channels here in the axon hillock. You have sodium channels. These are what are called voltage-gated sodium channels. And this input that comes in from the dendrites changes the voltage potential in the area of the axon hillock. And if it changes it enough in a certain direction, it will cause those voltage-gated channels to open, and it'll cause a depolarization of the neurons. So the neuron's kind of an all-or-nothing thing. It's just like the heart, where it either depolarizes or it doesn't, right? It's an all-or-nothing. And all of this information coming in, this sensory information, can either be excitatory or inhibitory, right? And they're, on the order of thousands of dendrites coming in, all putting excitatory and inhibitory information. And when the excitatory information predominates over the inhibitory, then it'll depolarize. So far, so good? Yeah, so when you calm yourself down, that would be inhibitory information going into areas of the brain, sending inhibitory information out to other areas of the brain, calming certain things down. Yeah, and we'll talk about the chemical basis of that here in just a little bit. This is kind of the big picture, but yeah. And then when that depolarization occurs, right, you have an action potential, and that action potential propagates down the axon, and the axons may or may not be myelinated. And then you have these little areas called the nodes of Ranvier, right, in between the individual myelin sheaths. And then at the very end, you have what is known as the axon terminal, and you actually have thousands of these little axon terminals. And these axon terminals then interface with dendrites of other neurons or receptors in organs, right? And where that interface occurs is known as the synaptic cleft, right? Now, the synaptic cleft, or the synapse, has a special name if the neuron is interfacing with muscle, with skeletal muscle. And that special name is known as the end motor plate, or the neuromuscular junction. The neuromuscular junction is nothing more than a synapse or a synaptic cleft in the somatic nervous system. So like we said, neurons, it's an all or nothing kind of thing, right? And it is the summation of all of the all the inhibitory and excitatory information that will make the neuron more or less likely to depolarize. When excitatory information predominates, the neuron depolarizes. When inhibitory information predominates, the neuron won't depolarize. And so it's kind of like a switch on-off. Only you have 100 million of these switches with 10,000 or so connections with all the other switches going on to make this all work. We know that in...
Question real quick.
Yes.
All the branches off the axon terminal,
Mm-hmm.
do they all give inhibitory or all
No.
excitatory? They can be.
Yes. So it's, yes. And in some contexts it's excitatory, some contexts it's inhibitory. And some neurotransmitters are excitatory in some cases and inhibitory in other cases, and it depends on which area of the brain that axon projects into. And I'm going to talk about neurotransmitters, but that kind of leads into what I was going to say, is that in the axon terminal, you have these little vesicles here filled with neurotransmitters, and then those vesicles will move out, right, through exocytosis, and release neurotransmitters into the synaptic cleft. Those neurotransmitters then will then attach to receptors on the dendrites of other neurons and will either result in excitation or inhibition depending on the type of receptor that they're attaching to and the type of neurotransmitter involved and the area of the brain or the specific neuron that...
But each branch is sending out the same
Not necessarily, no. Yeah, it is really convoluted. Yeah. It's like Alice, and it just, you go into Wonderland and it just gets weirder and weirder and weirder. Right. Yeah. Huh? Actually, the book, yes, is somewhat of an analogy for in one way of interpreting it is an analogy for a psychedelic experience. That's one of the common ways of interpreting Alice in Wonderland. Yeah. And they're like little like the the mushrooms and the caterpillar smoking, and so there are some there are many other ways No, it's just kind of a weird noise, huh? Anyway, yeah. I'll get you guys on a break here in a few minutes. But anyway, that's totally an aside, but yeah, we're like Alice going into the rabbit hole and looking and going, oh my God, this is a lot. Okay. Yeah. So each neuron may have axons that interface with approximately 10,000 or more other neurons. Lots of connections going on, lots of craziness. And you know what? Let's just stop here. Okay, cool. Well, let's move right along.
It's no picture.
Oh, yeah. I gotta plug this in, huh? There we go. All right. Hey, there we go.
All these things, they look like people that have been chopped up.
This is my drawing. My art's gotten... chopping people up. There we go. All right. So let's continue on. So we're going to zoom into the neuron a little bit more. All right. So here I have the soma here, and I have the dendrites. All right. And do you guys remember how depolarization occurs? Going back to cardiology. So you have a lot of sodium. Yeah, right. You have a lot of sodium outside of the cell. Okay. And you have a lot of chloride and potassium inside of the cell, right? And then you have two different types of sodium channels. You have your voltage-gated sodium channels. These are sodium channels that open up when a certain membrane potential is met, right? And then you have sodium channels that are attached to receptors that open up when a receptor activates them, right? Or they may be the receptor. So the way that this works is anything that lets sodium come into the cell is going to make the cell want to do what? No, sodium's positively charged. Right. Potassium's positive as well. It wants to what? Yes. Anytime anything lets sodium into a cell, right, that makes the cell more likely to depolarize, right? Because if you look at it here, I'll draw a little graph for you guys. Remember, the membrane potential tends to be negative, right? Tends to be down here negative, and then we'll say that this is 0 and this is positive, right? And the resting potential, that is to say that the charge inside of the cell is relatively negative compared to outside of the cell, right? So you have all this sodium outside of the cell, and that sodium wants to get into the cell, right? And I'll put quotes around once because it's not a conscious process, it's just chemistry, all right? So what happens is if I let a little bit of sodium into the cell, that potential is going to move up a little bit, right? Does that make sense? It's going to move up a little bit. So when a neurotransmitter attaches to a receptor and that causes sodium to enter the cell, it's going to move the cell up toward depolarization. And remember, neurons, just like cardiac cells, myocytes, have what's known as a threshold potential. So let's say that dotted line is the threshold. You guys okay with that? What does that mean? That means if I can get the membrane potential up to that threshold, if I can get it up to the threshold, it'll depolarize, right? But it will not depolarize until I get up to that threshold. Does that make sense? So, again, we're starting down here at our resting potential. Anything that lets sodium in makes it more likely to depolarize. And then anything that lets chloride into the cell will hyperpolarize or make it more negative, right? It'll push it down more toward the negative. Does that make sense? Because chloride is a negative, right? So chloride will hyperpolarize or inhibit the neuron, prevent it from depolarizing. Sodium will make it want to depolarize, quote unquote. Are you guys okay with that? Right? So inhibitory presynaptic potentials cause chloride channels to open. Excitatory presynaptic potentials cause sodium channels to open and sodium enters. You guys cool with that? What's that? Chloride inhibits, sodium excites. And remember, this neuron has 10,000 different attachments, different connections, and so there is a whole bunch of excitatory and inhibitory input coming into the neuron all at the same time. And what we call this is a summation. So when the sum of all the excitatory and inhibitory information reaches the threshold, okay, so right here is my threshold, and so I've got some excitatory and some inhibitory, some excitatory, some inhibitory, some excitatory, some inhibitory, right? It's right, all this excitatory and inhibitory information coming in. When the sum of it gets the membrane potential up to the threshold here, those voltage-gated sodium channels in the hillock of the axon open up, sodium rushes in, and the neuron depolarizes, and an action potential occurs. Are you guys cool with that? So, if I have drugs or processes that act upon inhibitory receptors in these neurons, that will cause more chloride to come into the neuron, more hyperpolarization, and it will depress the neuron, right? It will make it harder to depolarize. So here, I'll draw this graph. Let's say that this is the threshold, and this is the resting potential. And if I give a drug that causes chloride to enter the neuron, it will become hyperpolarized, and the potential will be way down here, which means what? I'm going to have to put more excitatory input into that neuron to get it to depolarize. It's going to be harder to depolarize. All right. The primary inhibitory receptor in the nervous system is something known as the GABA receptor, the gamma aminobutyric acid receptor, GABA. And GABA is actually a neurotransmitter as well. In fact, GABA is the primary inhibitory neurotransmitter in the brain. It's a primary inhibitory neurotransmitter. And we actually have drugs that work on these GABA receptors. The two primary classes are the benzodiazepines and the barbiturates. And you guys should be familiar with the benzos, because what are the benzos? Yes, so midazolam, lorazepam, diazepam, alprazolam, if you're talking about Xanax, right? These are all drugs that attach to the GABA receptor and make the GABA receptor more sensitive to the effects of GABA. So basically what happens is, if you give somebody a benzodiazepine, the benzodiazepine attaches to the GABA receptor, and then when the neurotransmitter GABA attaches, more chloride enters than would normally enter. Does that make sense? And that is how benzodiazepines cause central nervous system depression. Does that make sense?
So how does gabapentin fall into that?
It's complex, very complex, but yes, it does involve, in part, these receptors and some other things as well. Yeah. Gabapentin, or also known as Neurontin. You guys cool with that? Right? Now, if I give a drug that blocks sodium channels, right, well, that will prevent sodium from entering the neuron, and that will prevent the neuron from depolarizing, right? And this is what we see behind our sodium channel blockers you guys talked about in cardiology, right? If I inject lidocaine, for example, which is a sodium channel blocker, and I inject lidocaine close to a sensory neuron, what happens? So let's say that I have a laceration on my... What's numbing? What does that mean to numb? Yeah, so numbing prevents those sensory neurons from depolarizing, doesn't it? If they can't depolarize, they can't send information to the brain, right? Sensory information can't get to the brain, right? Does that make sense? And lidocaine just happens to be very sensitive to the specific types of sodium channels that we see in those sensory neurons. We have different types of sodium channels that we don't need to go into for this class, but some are motor and some are sensory and some are more or less likely to be bound by certain drugs. So what lidocaine literally blocks the sodium channels, right? And prevents, just like in the heart, right? Prevents sodium from coming into the neuron. And if sodium can't get into the neuron cannot depolarize, right? And therefore it can't work. Does that make sense? So we're already starting to understand how certain drugs work, right? And so we're starting to understand how benzodiazepines might be helpful if someone's having a seizure, for example. What is a seizure? It is chaotic, disorganized, random depolarization of neurons in the brain, right? So we give a benzo that makes those neurons more sensitive to the GABA, and more chloride enters those neurons, and it makes it harder for those neurons to depolarize, and so they're depolarizing less, which means what happens to the seizure activity? It decreases. Does that kind of make sense? How we're actually starting to put this together. Okay. Let's now talk about how that depolarization, that action potential, is transmitted through the axon. And it's transmitted through the axon via a mechanism known as saltatory conduction. Okay? Saltatory conduction. And saltatory kind of means to like leapfrog or to jump. So what happens is you have a bunch of sodium rushing into the axon hillock, right? And then that depolarization moves down the axon, right? That sodium does. But what happens is the sodium, because it's all positively charged, does the sodium want to be close to itself? No, right? The positives repel each other, right? Likes repel, opposites attract kind of thing, right? So the sodiums repel each other. What happens is as they start moving through the neuron, they begin to spread out. And so that action potential, that signal becomes weaker, right, as it spreads out, until we get to a node of Ranvier. And at the node of Ranvier, there are sodium channels that allow more sodium to enter, right? And so the sodium concentration increases and that signal gets boosted, and then it begins to spread out again, and then it gets boosted at another node, and it begins to spread out again, and rinse and repeat until it gets to the end of the axon. That's why we need to have those little nodes of Ranvier. That's why we need to have a chunk of myelin, a node of Ranvier, another chunk of myelin, right? And this is exactly what we see with like fiber optic communication, right? When you bury a fiber optic cable, that light kind of bounces around inside of there, and it kind of, quote unquote, spreads out, weakens out. So every so often, what do we have to do? Right? You have to amplify that, right, every so often. Well, it's kind of the same thing going on in the axon of a neuron. Does that kind of make sense? Now, if you didn't have the myelin covering here, what would happen? Well, that sodium would just kind of leak in and out and go all over the place, right? Just like if I were to take this power cable here, see this power cord? What would happen if I removed all the insulation to this cord and I just had the bare wire hanging there? It'd be a lot easier for those electrons flowing through that wire to go into other things like a human being if I were to touch it, right? Same thing here with those neurons. If they didn't have that myelin, you'd have more spreading out, you'd have ions going here and there, it wouldn't be as self-contained, it wouldn't be as efficient. And that's why demyelinating diseases can be so problematic, because you're losing that insulation. Okay, you guys good with that? Okay, so eventually that action potential makes it to the terminal axons, all right? And then there are voltage-gated calcium channels, and those calcium channels sense all that sodium, and that causes these calcium channels to open up, and calcium rushes in, and calcium helps these vesicles of neurotransmitters, helps them to dock to the terminal axon and release their neurotransmitters out into the synapse, right? And there you go. That's an action potential. Woo, huh? And that's just like the bare basic, scratching the surface of it. Cool. You guys good with that? All right. Good deal.